was a problem for many students to access the videos that I shared with you. The problem is that you should go to the Google all Yahoo, Hotmail, all of them were Gmail address, right? So in Gmail, go to all mails. All mails in the social category, right? For that one only you will get the videos. Right? All of them have been shared. I can share the screen and show you, right? All of them, again, it says that, again now, usually our class time is 6 to 7:30, right? So 6 to 9, right? Then why you may be wondering why that is. There can be half an hour changes due to the situation, right? I didn't have the power, still I don't have the power, right? And also the signal condition was so weak. Signal condition is weak. A lot of power cuts were there, right? So signal condition is weak. Still, it says that the signal strength is weak, right? I have installed all the antennas and all those things. Even with that, the signal strength is low. That is due to the low battery capacity. And when I'm having an extra class, right? Signal strength bandwidth signal strength, right? Right? To reduce the spreading of the news and all that, people's gathering. Reason still sometimes it says that signal strength is low. The power problem. So whole day I had some classes. 2023 batch, right? One class, and now 2024. So battery drainage was there. Now the power will come somewhere around 7. Then this will start to recharge again. That's why it took me some time. I mean, 6:30. Yeah, maybe let's somehow. Right? Right? But since this is a new class, it's not good to postpone for another day. Rhythm at least. But 2023 students are there, some of them. Just to cover the first things, you know that we have shift class with the agreement of both you and me. Temporarily, with the conditions in the country. No point of waiting at the queue, right? Mass polling right? Nothing to say about that one. I paid but don't know how to get the link. Watch the video. I will right. Come on, get used to it. Right. Okay. Then when I'm sharing the videos, live class when I'm sharing the videos, most of you have received the videos. No problem about that one. But they are received to your Gmail as a personal social category. Social category as a all messages you will get that one, right? And you cannot access with another. When you access to YouTube, you access with the email account, right? Note for today. Note copy. Note I think we only finished carbohydrates, right? Carbohydrate, protein, lipids, all of them. All of them are there. Even but somehow I put it, no problem. But if I think again, that's why I didn't bother to put the all of them are there. Yeah. That's why I didn't bother to put it there, right? In the previous class note, it is there, right? Okay, but I'll put it, it's not a new note. Those who have downloaded and have a copy. Have a copy means not the soft copy, hard copy, soft copy, yeah. Don't bother to download it again. Theory notes, periods, theory note 4, I guess. All right. Yeah. Okay, I'll upload that to the group. It will take some time to be uploaded. Now it's okay. Right, let's start. Right, page number 3, we have to start. Glycosidic bonds as I explained how the glycosidic bonds are formed. You checked your Gmail twice and didn't receive last week recording. NUSH Baduti. Okay, I'll check it again. Right, but Mama share kara. NUSH Baduti. Right, let's see Mambala right. Just check it 4 times, 5 times, 10 times. In the since you are new, no problem, right? Since you are new, get used to these things. How to get the link, right? This is the first time you are joining the class with the new link. Link from the Teaching First website. It's okay, right? Learn how to get it. This is the first time, second time. First time. I have shared these messages to the email account. So get used to that part. Come up now, right? Nan Badruddin, I have not shared it to you. It has not been shared to you. Oh, now check it whether you have. I have. Right? Then there was something, right? There can be problems. Then give me another. So it says it has been shared to you, right? Yeah. Now check it. If not, done. Right? It's okay. Now let's go to the more to the lesson. Right? Why I am taking time in the class itself for those things, because that is really important, right? For the whole 2 years, the same procedure will be applied, right? But I don't think that will be a difficult process, right? Tell me if you have such a trouble, no? Just like the theory parts, learning how to get the link from the Teaching First website. Video everything, how to register and all that, right? How to get the link from the Teaching First website and how to get the messages, private messages, that means the videos, right? All the things are right. Okay, yeah, now it's okay. Important character, it's a component of the class, that's why I do it in the class itself and finalize it and teach you how to get it. Very important that 2023 students, I don't have any problem, right? To get the following, previous notes, there is no any procedure. Give me a second. There is no any process to get the recordings. Take a message. Recordings I'll send you with your email, right? Email past notes. Past the group description, I think I have updated. Right? Go to the WhatsApp. When you go to the WhatsApp, in the WhatsApp there is something called group description. WhatsApp group description update. Right? Payment method I think I have updated. Usually specific number of specific number characters. Yes, no worries. Right? So I will share this one and show you, all of you. If you don't have the notes until the 13th note, so all the notes are not given here. Up to some extent, you can Google and plant until the 3rd lesson notes are there. 3rd lesson notes, this one, this one. Google WhatsApp. Never WhatsApp page in So in WhatsApp groups, you can get the group description now. I have updated the group description, right? Right? So theory notes, here there is a link, you can find it. Theory notes link WhatsApp group theory notes. There's a link like this, right? When you double click on this one, right, you can get the theory notes. So definitely you got theory notes link here, right? Likewise, not all the theory notes, most of the theory notes, I mean, for you too much. First lesson theory notes right? 16 theory notes are there. You can download all of them. Let me be in it, right? Is it clear for you? You can get all the theory notes from this link, right? Time to time, I'll be uploading, updating them for you. All the notes are there. Notes you can download it and have it. Is it clear for you? Any other question? Now let's start. Ready? Right, okay. So in the last class we learned that lipids is a theme. We have to start from I WhatsApp video call right. Page number 3, right? Functions of carbohydrates. So carbohydrates, we learned that carbohydrates are the same carbohydrates that you learned in all levels, but there were some new things. What are the new things? In the previous class, you didn't know. I mean, previous class means in the previous grades, I mean, all levels, you didn't know how the monosaccharides join with another monosaccharide. In the previous class or in A levels, we learned that one. That is by forming a specific bond called glycosidic bond. Glycosidic bond is a very interesting thing. A water molecule gets removed from that one. You don't need to learn the molecular structure or anything, but I have taught you everything. We have drawn that one, everything, right? But you don't need to know the specific type of bond. If it is two monosaccharides, one monosaccharide, the other monosaccharide, they join with each other and forms a disaccharide by a glycosidic bond. A glycosidic bond is there. Glycosidic bond. Not only for disaccharides, but also polysaccharides also. Monosaccharides join with each other using glycosidic bonds. We discussed about the glycosidic bonds, how they are formed and all that. Thereafter, we came to a very important part, that is oxidizing sugars and reducing sugars. Oxidizing sugars, reducing sugars and non-reducing sugars, right? So how that happens? What is oxidizing nature? Oxidizing oxidation, right? What is oxidation? What is reduction? How does it occur, right? And we looked at the Benedict test with a different perspective. Benedict test precipitate. Now you learn copper sulfate, Cu2 plus ions are there. They are oxidized, right? It goes under oxidation, right? Cu2 plus cuprous forms cupric Cu ion. That is brick red in color. That's why your brick red precipitate is formed. Nicely we learned all the processes. Those who didn't learn, get the video and learn. I have shared the video. So we have finalized it. Those things we discussed, right? So monosaccharides, disaccharides, and polysaccharides we finished. Up to this level, you thought that you only knew 3 polysaccharides, right? Starch, cellulose, different polysaccharides are there with different monosaccharides molecules, right? You learn new things. So right? Okay. Again, it says your internet connection is unstable. It is not a problem of mine. Again, still, it's like I told you, powers are switched off. Then right? So class start current charge, but until then, if you have any problem, if you cannot hear me, if you have any problems with the video, tell me. Message then I can repeat to do something about it, right? Okay. I'm not watching with another device, but our device is key. If you have any issues, send me a message. Right. Right. Then functions of carbohydrates. Easy, but it is here also. We are going to look at it with a different perspective. Right. Previously, we just learned, right, without considering whether they are monosaccharides, disaccharides, or polysaccharides. So, Oliver, can you remember? We just discussed 5 functions, right? They are storage components, right? Gives energy, likewise. But now, here we learn them as monosaccharides, disaccharides, and polysaccharides. Specific functions of different types, right? Monosaccharides. Well, messages usually make some big link, some big bin. Right. Okay. Monosaccharides, energy source. Obvious for you. Energy source means a source which can produce energy by undergoing, yes, respiration, cellular respiration. Cellular respiration. Right. Okay. Glucose. Right. You know that one. Anybody know levels you learn. How does it happen? If you prefer, you can write it here. But later in this lesson, we have to discuss a lot about the respiration. You can decide, but you know about the respiration. How does the cellular respiration occurs? Well, message is done, yeah? Make it. Glucose molecule. This is all level knowledge, right? But it's okay, not wrong. Plus an oxygen gas forms energy plus carbon dioxide plus water. If you just recall, this is the exact opposite of the photosynthesis. Photosynthesis is carbon dioxide and water with the support of light energy form glucose, and oxygen was formed as a byproduct. By you. Right. Anyway, is it clear for you? This is the respiration. So glucose molecule participate in the formation of energy in the form of ATP. Right. So energy source. Building blocks of disaccharides and polysaccharides, that is obvious for you. I'm a right. Then components of nucleotides. DNA is formed of, later we are going to discuss, deoxyribonucleotide by the multiplication of a molecule called deoxyribonucleotide. The sugar, pentose sugar, is deoxyribose. RNA, ribonucleic acid, is formed by the multiplication of another nucleotide which is known as ribonucleotide. RNA. Ribonucleotide. Right? So the pentose sugar for this molecule is provided by the carbohydrate. Since the pentose right? Triose, tetrose, pentose, hexose. Those were. So they be monosaccharides depending on the number of carbon. Hexose recall previous week, right? This is a hexose. 6 sugars are right. This is a pentose sugar, ribose and deoxyribose. So pentose sugar, monosaccharide is present here, right? The monosaccharides, right? Functions. Any question? You know how to get the tutorials. Power cuts the night in the way. Class Then disaccharides, they will join. Disaccharides. Storage sugar in milk, lactose. Again, storage component in milk. Then translocation in phloem. Now, we have to discuss it separately in the plant, but it's a very interesting thing. Actually, if you did all levels very well, still I repeat. Right. Please read the O-level science things, not only the biology things. Actually, for all the other physics and chemistry also, this is important, right? Please read O-level science. O-level science activity stick or if you like do the past papers even. Be well versed in O-level things. O-level, they were very well versed in. O-level. If you like, you can teach some O-level students, right? You can teach an O-level student, right? So somehow upgrade the O-level knowledge. If you have done O-levels very well, you should remember this relationship. It's a very important thing. O-levels for my students, I have to taught this one, right? There's a question. Right, okay. 7:15 I can give you, right? Right? I will give you a small break. This is the relationship. You should remember. The type of carbohydrate produced in leaves was glucose. Glucose. Produced. It was immediately converted to starch in the leaves. In the leaves. Temporary storage. That's why we were able to do the starch test to identify that the photosynthesis has occurred. Can you remember grade 11 second lesson? We proved that the photosynthesis has occurred by doing the starch test. Starch test occurred. That the reason is, as soon as the glucose is formed, they turn into starch. Starch. Temporary storage in the leaves. But leaves cannot store a lot of starch, so they should be transported. Now, my starch is not water soluble. Previous week, under the physical characteristics I told, right? They are not water soluble. So, in the plants, these materials are transferred by dissolving in water. Xylem, phloem, these are transferred in phloem. But in both xylem and phloem, materials are transported by dissolving in water. Minerals are transported in the xylem by dissolving in water. So these materials are transported in phloem by dissolving them in water. So they should be dissolved in water. For that matter, they should turn into the disaccharide. Why not the monosaccharide? If they are turned into monosaccharides, I had from the beginning itself. Disaccharides are 1,000 molecules if they are made into disaccharides, 500. Then new 500 bonds should be formed. If they are turned into monosaccharides, a new 1,000 bonds should be formed. Right? Okay. So the plant is tricky, right? So they are turned into disaccharide with the aim of dissolving in water and transporting. So they are converted to sucrose now. Sucrose. This is transported to the particular place which it gets stored. Sink source relationship under the plant, right? But you know these things in all of us. Then sucrose is transported to that particular place through the phloem and stored there as starch final. This is the final storage. This is the relationship, right? So this is the place where it gets produced, production. This is the temporary storage. This is the transportation. This is the permanent storage. This happens in the leaf mainly, yeah. Whatever green color part can do this. This is also at the leaves mainly. This is in the phloem. This is, we cannot exactly see a place. This is coming later. root දැරට stem or fruits. When we are sink source right later. Should remember this relationship. Actually, if you want, go back to the whole. So this is where that's sucrose. Sucrose, the fact that we discussed. Translocation in the phloem. Transportation, translocation, correct? In the phloem, sucrose. Or now whether translocation translocation, phloem. I like if you can write this. Officially, we discuss it later. Where to write? Wherever you have the space after writing these functions of these things. Yeah. Correct, huh? What do we call this process? This is not an exact process. This is what actually happens for all the food that is produced inside the plant. There's no exact process, right? The transportation or translocation process is taught to you how it happens, right? All those things under plant as the phloem translocation process, right? Phloem loading, unloading, sink source relationship, likewise. This is what happens, right? This is the fate of the food produced in plants. Plants, right? Are there any students who obtain good island ranks in the biostream? Again, oh, the bio again. My class again, are there students who obtained a good rank? Students who obtained a good island rank in biostream. Biostream make the island 1, 2, 3 කියලා තියෙනවා. English medium ළමයි ඉන්නවා කියලාද මගේ ළමයි ඉන්නවා කියලාද නැත්නම් island ranks are given for subject wise, not like O levels. O levels are the island biology world island first skill. Chemistry physics for biology stream make it island first skill. For mathematics stream, island first skill. Then a technical stream, commerce, arts, right? Physics or resource book at the English medium. Single medium. Do you have the single medium one? English medium, Mama Dapus, Hunda, get look at the English medium physics. Physics single medium, English medium. Come a little liberal than I was not that much worried about the. Yeah, I will get about that. There are. Okay, I look into several teachers, some other teachers. Yeah, monkey, but that depends. Chemistry, physics really depends on the person. Are subjective case. So really subjective case. Physics resource book, Darasiki. One soft copy gati. Whatever no chuta. Internet in now, student Lanka physics resource book here. UGC handbook whenever possible, them upload karana. Mang itune picture upload karna tamara onna mate explain karana ma UGC handbook padikiyanu ma explain karana athara mate barna in the introduction I forgot. I did a session with Tigama Single Kalati Nikamam Karnate Mama up a university got thicker. Ma Ogulunda Net Mogulu single medium American explain I have done it. That was done in single medium, right? And I couldn't get the chance to explain a lot of things about my stream. Limited time was given. It was better if I had the chance to explain it here. I will upload both the books. If you have any problem about UGC handbook, right? It's good. It's good to have a target. It's good to have right? UGC handbook 2024 students. I'll upload that, right? Meeting වැඩත් එක්ක 23 ළමයි විතර ටික දෙනෙක් ඉන්නවා මම හිතුවා තරහ වෙලත් ඇති කියලා ළමයි. ගොඩක් ඒවා අප්ලෝඩ් කරන්න වෙලා ගියා. වෙන මොකුත් නිසා නෙවෙයි මේ රටේ with the condition that exists in the country. දැන් ඕගොල්ලෝ හිතලා බලන්න ඕගොල්ලෝ මේ කිව්වත් එකක් බලන්නේ අරගලේට මොකද උනේ. ඇත්තනේ මොකද්ද මේ රටේ තියෙන සිටුේෂන් එකක් බලන්න ඕනේ. මම ඒකේ. ඒත් එක්ක අපි ගොඩක් දේවල් වෙනස් වෙනවා අපි. Like lot of things change. Actively වැඩ කරන මිනිස්සෙක් හිතන්න කොච්චර අසර වෙනවද කියලා ඒ වගේ condition එකක. හෙට අදන plan එක මොකද්ද කියන එක අපිට අද හිතා ගන්න බෑ. හරි. You have both of them, both the languages, right? Sinhala and English. If you want me to describe it, group next class, right? Or send me a message to the group, right? As a comment for this book, comment tag make upload. If you need an explanation, please explain this one, that one, likewise. This is the latest version. Send me a message. It will take some time for them to upload. One of them is 33 megabytes, the other one is 6 megabytes. Right? I don't know why that is 33 megabytes. Single handbook. English handbook word copy single handbook photo. Right. Both of them are there, they will be uploaded. if you have any problem with that one, if you want me to explain anything related to that one, ask me as a comment or just send me. I want an explanation about my stream likewise. Actually, the first day I didn't refer this. It requires a lot of time. That's why I didn't do. But if you want, right, I'll explain, because now we have done all the explanations. Now you know how to work in my class. Right? Tutorials, notes downloading, sharing the videos, registering to the class. I have taught you all the things, extra stuff, right? So we have some time to allocate for the discussion of this one if you want. So add that as a comment. If you want, now both the UGC handbooks are uploaded to the group. Hi. Hi. Okay, it's good. Okay. That's good. You talk to me. Oh, YouTube video live. Oh, YouTube live YouTube live cannot be watched. Recordings. Recordings. Is it the problem about the recordings? To watch the recordings, you should access YouTube with whatever the email that you have sent me. Now this live stream cannot be watched in YouTube. It is private. I should share it. I should allow the access for you to watch it, unless anyone else cannot watch. Right. YouTube the live no need to watch the YouTube. Okay. Physics handbook. I look into that one because I have lectures about program that was done in English medium, single medium, right? That's why I didn't bother to share it to you. But he did a program, very good one. But I wasn't able to express a lot of things because I had limited time of 10 minutes. Right, okay. Yeah, English medium students, they have very good ranks. Yeah. Very good ranks are there. But medical faculty right? The English medium man got selected to medical faculties. A lot of students are there, right? No problem about that, no doubt about that, right? So, then let's shift to the next part. Translocation in the phloem sucrose, storage in sugar cane. All right? Most of the times, right, permanent storage happens as starch. Even the temporary storage happens as starch. But in sugar cane that we use to produce sugar. In sugar cane, if you eat, all of you may have eaten sugar cane, right? At least once in your life. Then you feel that sweet nature. Sugar cane, sweet nature. The reason is, in sugar cane, it is stored in the form of sucrose. Maybe they don't store in special case. The special case. Usually in plants, carbohydrates are stored in the form of starch. Starch is the stored. But in sugar cane, the special case is stored in the form of sucrose in the stem. Special case. This is disaccharides. Polysaccharides, storage polysaccharides. In both plants and animals, storage polysaccharides. Carbohydrates are stored in the form of polysaccharide, and in plants, that is starch. In animals, that is glycogen. Both of them are O-level knowledge. They are not glycogen in muscles and liver, right? Only prayer time, right? Quickly, 7:30. 7:30 come right to 7. Come before 7:30. 7:30 sharp at 7:30. Yeah, right over 7:30. That is going to be the break also for you. Break until then. Now I think for some students need some time for the prayers. I don't like to give a break. Come up with break. Others try to access it's almost uploaded. Sharp at 7:30. Then 7:20. Sharp at 7:30. Let's join. Disconnect If you have any question, you can ask me. මොනවා වෙන ප්‍රශ්නයක් තියෙනවා මම listen කරන්න. After 5 minutes, I'll start answering him. ගොඩාක් ලොකු break එකක් ගන්න බෑ. Thereafter continuously I'm going to conduct the class. That is for the prayers එක. සාධාරණ හේතුව. හරි. ඉතින් අනිත් අය වතුර ටික කොම් කියලා ready වෙන්න. Let's start sharp at 7:30. right? There's a question: is there a book called biology practical පොතක් තියෙනවා? I think the practical book is also there. නේ practical after finishing this part we have to do the practical. පොත දාලා නැද්ද බං? I put it right. Practical book is there. Oh, practical පොතක් තියෙනවා. You don't need to buy it. I have a soft copy. I will send it to you, right? Soft copy is මම දාන්නම්. Physics also has then we go until you have the break, right? I was looking for that one. Physics වල තියෙන මේ පොත. I didn't have all the copies. You get the copies in the physics book, right? Resource book, advanced level, right? National Institute of Education, right? Is it? Other units වලටත් තියෙනවා මම හිතන්නේ. Unit 1 2 3 නම් මම ඔය කියන්නේ. Unit 5 6 7 10 11 10 10 ක ටයිම් වලින් තියෙන්නේ 5 6 7 8 9 නා උඩින් තියෙනවා resource in English. මෙන්න මෙතන ඇතියා බලන්න මේ සැරේ කිව්වා මෙතන look at the downloads, right? At least if one of you have shared with the others, biology group එකේ කමන්න share කරගන්න. හරි කමක් නැහැ. check කරලා තියෙන්න ඕනේ. Did you join the protest කියලා? No, I am not in the western province. නැත්තම් මම ගිහිල්ලා මෙතන. හරි. එහෙනම් class එක cancel කරලා ගිහිල්ලා. කරලා ඉවරයක් කරලා දාන්න ඕනේ. හරි. ඊළ කහලා මෙල්ල වල දාන්න. නිකන් කතා කරලා ඉතරන්නේ ඔයගොල්ලොත් එක්ක. හරි. Okay, let's continue the class, right? Okay. So polysaccharides, storage polysaccharides, just like I told you, glycogen, that is for animals, and fungi, starch, that is for plants. Inulin stores fructose as energy source in Dahlia tubers, right? Inulin is storage in Dahlia tubers, right? Then structural polysaccharides. Some polysaccharides participate in formation of structures, different structures. For an example, plant cell wall is from cellulose. Hemicellulose killer part secondary wall. Let's discuss it later. Cellulose in the cell wall of plants and chlorophytes. Pectin in the middle lamella of plant tissues. I explained you what the middle lamella is. When two cells get together, these are things that we have to discuss later. That's why I'm not going for deep explanations, right? When two plant cells bind with each other, there is a glue-like structure called middle lamella. So if this is the first plant cell wall, if this is the second plant cell wall, in between them, there is a structure, there's a chemical called pectin, which is composed of calcium pectate and magnesium pectate, right? That is also a structural polysaccharide, pectin. Dahlia කියලා කියන්නේ plant එක මට පෙන්නන්න වෙනවා නෙවෙයි. Since there are some Tamil mediums, Tamil Sinhala නම් කියලා හරියන්නේ නැහැ. Right, I'll show you the plant. මෙන්න මේ plant එක. හරි. මේ ඔයගොල්ලෝ ෂෙයා කරන්න බෑ පොඩ්ඩක්. මෙන්න මේක right. පස්සේ බලන්න ඔයගොල්ලෝ ෂෙයා කරන්න. මේකට ගෙවල් වල සාමාන්‍යයෙන් මේක එකම නෙවෙයි ඩේලියා කියලා කියනවා. ඩේලියා කියලා කියනවා right. ඒ වුණාට ඒක වෙනස් වෙන්න පුළුවන් varieties right. ඔයගොල්ලෝ මේක අහලා තියෙනවා very similar plant which is yeah එකම variety එක වෙන්නත් පුළුවන් මේකේ lot of varieties are there right. මේකේ tube එක ඩේලියා කියන එකත් ඔයගොල්ලෝ ගෙවල් වල හදලා තියෙනවා නම් තියෙනවා දැකලා දෙකේ තියෙන tube එකක් the rhizome right that tube. I don't know what is it called in Tamils and all the others. Right? Then structural cellulose, sorry. Pectin in the middle lamella, you know. Hemicellulose, lama again, not only cellulose, but in all levels you learned that the cell membrane, the cell wall is composed of cellulose, but there's another chemical which is called hemicellulose. It is present in smaller quantities in the plant cell wall. Then plant cell wall abhi thayin cellulose vitarai inno. Hemicellulose. Passe lignin kiyala jathikuth thena. That forms the secondary cell wall, right? So, hemicellulose in the cell walls and plants. Peptidoglycan in the cell walls of prokaryotes. Prokaryotes. Bacteria wage cell wall එක. Peptidoglycan. If you take a bacteria, cell wall of them is also composed of, they also have a cell wall that is composed of a chemical called peptidoglycan, which is a polysaccharide. Then chitin, chitin කියලා එහෙම කියන්නේ chitin කියලා කියන්නේ right. Chitin in the cell walls of fungi and exoskeleton of arthropods. I explained that one when I was explaining about the polysaccharides name. Chitin ගැන කතා කරා right. Okay, with that we wind up the session for carbohydrate session. Not the class. Any question for carbohydrates? If not, we are going to quickly shift to the lipids. Any question for carbohydrates? Any question? Right. Is the liver only place? No. And muscle cells, right? Muscle cells also store. They go Khana නැති කෙනෙක්. ඉතියෝපියා වගේ කෙනෙක්. ඉස්සර අපි khana නැතිව ඉතියෝපියා. දැන් ලංකාවේ khana. Ethiopia මිනිස්සුත් example එක ගන්නදී ලංකාව. Such a person, such a child, how do you recall that structure of the body? Bones and the skin. Like විතරයින් තියෙන්නේ no muscles. That means for the growth of muscles, the food that we take, the excess food, right, is converted to glycogen and they are stored in muscles. If you lack or if you reduce the amount of food that you take, right, there are different methods that you can get fatter or thinner, right? One method is the amount of food deposited in the muscles will get reduced and you will become thinner, skinnier, right? The other thing is fat layer is deposited. Endodermal fat layer is there. The fat is deposited, right? But muscles is the second place. Muscles are deposited, right? Then let's move to the lipids now. So when you move to the lipids, again very simple. First we discuss the O-level knowledge. Lipids also have the same structure or same actually elemental composition, right? CHO elemental composition is carbon, hydrogen and oxygen, but previously in carbohydrate we observed 2:1 ratio between hydrogen and oxygen, but here we cannot observe that relationship. So CHO is present, that's all, right? Lipids are hydrophobic molecules. Hydrophobic molecules means the molecules which do not like to get involved with water, right? If you can remember O-level knowledge, right? Cell plasma membrane, when we discuss about the cell plasma membrane, you learn that it is made up of two phospholipid layers. We call it as the phospholipid bilayer. Bi හැමතැනම වෙනවනේ. බයියුන්ගේ අන්තිම කාලේ ඉඳ වෙන්නේ. හරි. Bilayer. Bi කියන්නේ ඇත්තටම ලැටින් වලින් two කියලා කියන්නේ. වෙන bi එකක් නෙවෙයි. Right. Phospholipid bilayer. In that one also hydrophobic tail කියලා එකක් අපි දැක්කා. Hydrophobic. Don't like to be with water. There are large biological molecules. They are large biological molecules, but they are not polymers or macromolecules. Very important to remember. In carbohydrate, we observed all the types. Smaller molecules, which are monosaccharides, somewhat larger molecules, right? And very large molecules. Right? But these are large molecules, but they are not polymers. Then how they are composed of? Right? There are three main biological important types of lipids. They are fats, phospholipids, and steroids. Let's look at them later. Right. Number one, fats. Fat is a type of lipid. Fats are made up of glycerol and fatty acids. So again, there is the structure. I'll show you the structure and I'll draw it. But for now, I have to change the power source. Right. Okay. Again, I'll show you the structure which is available on the book. UGC handbook upload. If I want to explain anything, add a comment. I'll explain it next week. Today we had to use the time for a lot of unnecessary things. Right. Unnecessary. But apart from the lesson, whatever we discuss is kind of unnecessary. Right? Again, you don't need to learn this structure that I'm drawing now, but if you are having any clues or doubts, you can use that knowledge, right? Right. This is the structure, right? Before sharing this, I'll draw some structure for you. So Then I'll go with the letters and all that. Now, this is composed of 3 molecules. Lipid is fat is composed of two types of molecules, right? 2 types of molecules. One is called the glycerol. Glycerol, 1 molecule. Glycerol molecule joins with 3 fatty acids and forms a lipid. Remember the lipid kilometer, right? Lipid plus 3 water molecules. This is how it happens, right? I'm going to draw it with symbols. Symbols, right? Glycerol is comparatively a larger molecule. Glycerol is a liquid, larger. Glycerol. Glycerol. Then fatty acids, they are like this. Fatty acids are like this, right? It's like this. 1 glycerol joins with 3 fatty acids. So now this molecule is called a lipid. While this forms, 3 water molecules are separated from these places. H2, like this. Is it clear for you? Right. Now let's look at the molecular structure. Right. I'll show it. First, I'll share it and show it to you. Then let's draw it. This is the structure, right? Again, there's no need for you to remember this structure. This is the glycerol molecule, right? It's the glycerol molecule. Glycerol molecule. And the 3 fatty acids. The fatty acids too. Right? All together, they have formed now, right? The structure. Now the value. Right. Now I'll show you again. Huh? I draw it and show it. The blank is given there for you to draw this. Not to draw this one, but let's draw it, right? Glycerol means something like this. C, right? Don't draw it. Still don't draw it. H, you know that carbon forms 4 bonds. Hydrogen, oxygen, and hydrogen. CH2O is clear. Then carbon, hydrogen. The glycerol. It has been written organic chemistry it can be written like this. CH2 but I have depicted, shown it in the way that we can show the 4 bonds of this one. Are they clear? Glycerol, is it clear for you? Right. Then fatty acid. Here R hydrocarbon group. This can change from fatty acid to fatty acid. Any number of hydrogen, any number of carbon, right? Hydrocarbon group. C2H5 likewise same right? C double O right? R means different group. Now what happens here is, Lamai, carefully draw it. Right? Right. These are the molecules. H2O. H2O gets removed. Draw this line. This structure. That's why I have given you some space. Huh? Even for the examination, it won't be asked. Right? Draw it. Ester bond. The bond which is formed here is known as ester bond. I have drawn the ester bond. Right? Ester bond. Ester bond. Right. Draw it quickly. Can you see? Yeah. If you like, you can draw this structural thing also. Right? I will clean this for you.
The lead sum
Yes, this structure is the lipid molecule. Yeah, this structure is the lipid molecule, right? Now I'm going to erase some part. That's why I gave you time and I waited, right? So here you can see when a water molecule is removed, no water molecule like removed. Likewise, all the water molecules are removed. Right. Then, since here is an electron and here is a free electron, a bond is generated here. I'm going to exaggerate. Usual bond just like this, but I'm going to exaggerate that one. Special case, otherwise it's just like chemical bond. Here is an excess electron. Here is an excess electron. They covalently bind with each other. Normal, right? But since I want to exaggerate that bond, I'm going to draw it different. This bond is known as ester bond. Previously in carbohydrates, what did you see? Glycosidic bond. Now ester bonds on a new terms. Definitely you should know for the A levels. Ester bond. Ester bonds, right? Right. Right? You don't need to erase and draw it, right? If you want at home, separately put an arrow and draw how this happens. But when a water molecule is removed, you can just know. And fluent is a bond form. Ester bond, right? Right. Let's move to the note. So the space was given for that part. Glycerol belongs to alcohol group having three carbons and each of them bear single hydroxyl group. Previously observed. One glycerol, 1, 2, 3 carbon, and previously each of them possess hydroxyl groups OH minus, OH minus, OH minus. Fatty acids are hydrocarbons containing with long, that means 16 to 18 carbon skeleton with a carboxyl group at its one terminal. Here, R this part is a hydrocarbon group which compose usually 16 to 18 carbons, and this is the hydroxyl group here. Fatty acid molecules bind to each hydroxyl group of glycerol by ester bond, resulting in a fat molecule. Fat molecule is the fat molecule, right? Name this is the fat molecule, final product. Glycerol and fatty acid if you want. Then a lot of other diagrams will begin. Images but otherwise someone like terms when you do the organic chemistry nothing right? But until you do the organic chemistry organic chemistry are lazy, right? In organic chemistry, these are things you have to learn. This is a glycerol and hydroxyl marker that you want hydroxyl, right? Fatty acid carbon. This is the hydrocarbon group R, which contains 16 to 18 carbon, right? Again, there is a hydroxyl. මෙයා තමයි lipid කියලා ගන්න. right? right. මේකට triacylglycerol කියලා කියනවා. Triacylglycerol. Triacylglycerol. I'm back to right now just to see this glycerol molecule. Yes, there can be differences, facts, but almost same කියලා ගන්න පුළුවන් for you. No problem. Yes. Triacylglycerides and fats are same here. Yes, that now. Hydrocarbon chains, R group, right? Of fatty acids contribute to hydrophobic nature of the fats. Based on the nature of the hydrocarbon chains of fatty acids, they are categorized as saturated fatty acids and unsaturated fatty acids. Right. 16 to 18 carbon. 16 to 18 what kills the carbon. Carbon chain. Right. Now we want to talk about this carbon chain, right? Right. Now this carbon chain of this, right? Carbon 16 to 18, right? So in between these carbon, hydrocarbon, R group. I'm talking about the R group now. If there are double bonds now, double bonds like this, here and there, one or many, right? Like this, goes like this. If there are double bonds, the meaning of that one, right? That's how the names come. Huh? If there are double bonds, these double bonds we have carried bond. Right? 3 bonds are there, then hydrogen. These are hydrocarbons, this hydrogen. Hydrogen. Here 2 hydrogen molecules. Because carbon can form 4 bonds around it. That's the nature of carbon. How many? Right, you can see, right? Since there are double bonds in between carbon, what can they do? They can join, or they can combine with other molecules like this. This is the original structure. They can join with other groups. I'm going to denote them X and Y. I can break this bond and X group within join because carbon can form 4 bonds, X and Y. For this double bond also, A, B. Simple chemistry, simple. Solar. Right? Saturation grade 11, third lesson, right? When you have dissolved the maximum amount of things that can be dissolved, it was saturated solutions. Thereafter, if you dissolve excess amounts, they start to deposit. Maximum number. Sugar, there's a maximum number. Saturation. Grade 11, third lesson, right? That's why I told you. Grade 11 science. Saturation means maximum amount. The opposite of that one is unsaturated. Unsaturated means it has not combined with the maximum amount of molecules that it can combine. Mean that's saturated. Now it has combined. Can I, if I want to join some extra molecules, I will have to remove an existing one. So it has joined the maximum 1, 2, 3, 4, 5, 10 molecules. So in the R group, R group means this one. In the fatty acid, in this R group, right? Hydrocarbon group. R can akura. It is representing hydrocarbon group. In this hydrocarbon group, which is composed of 16 to 18 hydrogen and carbon, so carbon, 16 to 18 carbon. If there are double bonds, just like my previous case, they can break down. Double bonds can turn into single bonds and join excess molecules. So that is known as unsaturated fatty acids. If they have single bonds like this, that means all the places are filled. It cannot join furthermore. So they are saturated. The maximum amount has been filled. So if double bonds are present in this carbon chain, it is known as an unsaturated one. The reason is just like in the previous case. Double bond. Unsaturated because the maximum amount of things are not combined. How could I break this bond? Two excess things can be joined. Unsaturated. Likewise. Unsaturated and saturated name comes like this. But for you, it's sufficient to know if double bonds are present, that is unsaturated fatty acids. If only single bonds are carbon-carbon single bonds are present, that is saturated fatty acids. It's a question. Yeah. First lesson you get the have extra knowledge saturated and unsaturated. Right. Grade 11 first lesson. Fats are made up of saturated fatty acids. Fatty acids with hydrocarbons having no any double bonds, usually animal fats, come under this category. They are mostly solid at room temperature. Example, butter. Single bonds nothing. Saturated meat. Maximum amount has been joined. I beg right. Unsaturated fats. Fats made up of unsaturated fatty acids with hydrocarbons having one or more double bonds. In my example, there were two double bonds, right? Here and here. Usually plant fats come under this category. They are mostly liquid in room temperature, right? Saturated liquid, unsaturated liquid. Saturated means single bond, right? In here it is the meaning, but saturated means maximum amount. Saturated line, right? Maximum amount of solutes are dissolved, saturated solution. Maximum amount of things are combined, saturated molecule. Likewise, that is how the name comes, yes. But here, if single bonds are present, they are saturated. Right? So this is a saturated fatty acid. Yeah. Saturated one, and I'll draw an unsaturated one here. R1, R2, R star. One, two, three, four. 1, 2, 3, 4. 1, 2, 3, 4. This is an unsaturated. Huh? Right. So some questions are there, right? Okay. Generally, at what time period the entire biology syllabus must be covered before the exam? There is no such thing, nah? But before 6 months I cover. කොහොමද දන්නේ? 2022 ඒගොල්ලෝ නම් cover කරලා තිබ්බ ඒගොල්ලෝ ගැන කතා කරලා ඒගොල්ලෝ confused වෙලා ගොඩක් බය වෙලා 2022. 2023 students නම් මෙතන ඉන්නවනේ නේද? ඒගොල්ලොන්ට I have covered more than exactly half of the syllabus. Then fifth lesson එකේ භාගයක් කරලා භාගයකට 30% වගේ කියලා 40%. Fifth lesson එකේ අමාරුම එක සහ the longest lesson which requires 160 periods taking 40% වගේ ඉවරයි. දැන් 2023 තාම විභාග අවුරුද්දවත් ඇවිල්ලා නැහැ, right? Even if the examination is held in August of 2023, obviously ඒකේ ළමයි විතර ඉන්නවනේ. උගලට හොඳටම sure නේ අපි ඉවර කරනවා February එනකොට ඉවර කර ගන්න පුළුවන්. We can finish before February, right? No problem. මම මේක August September වගේ ඉවරනකොට ඔයගොල්ලෝගේ animal part එක ඉවර කරනවා. I'm talking about 2023 students, right? So nothing to worry about. Before 6 months we definitely finish. මාසයකට කලින් ඉවර. ඊට පස්සේ මොකද කරන්නේ? නටනවා. නෑ. Then past papers are discussed with you. Third term test එක කරනවා grade 11 third term test. Thereafter we are going to discuss the past papers with you. ළමයි to discuss the past paper it requires somewhere around 3 weeks. සති 3ක්වත් යනවා. Because discussing doesn't mean just giving the answers, right? Lot of things are there to be discussed in relation to past papers, right? Actually we predict කරනවා කියලා කියන්නේ guess කරනවා නෙවෙයි. Yeah, we predict with the style and all those things. 75% of the paper we ඔයගොල්ලෝගේ අවුරුද්ද කවදා වුණා whatever 2023 2024 75% in a ප්‍රශ්න ටික මෙහෙම හදාගෙන යන්න, right? Because we can do this can any teacher can do this when they follow the syllabus and the questions. දැන් ඔයගොල්ලෝට O level කියන්න පුළුවන් නිසා මන්නේ if you did O levels very well then O level කරපු ළමයි නා, right? When you look at the syllabus and when you look at the past papers කවුරු මේ පාර අහනවා 2022 student question from this lesson there's a question, okay? හැම පාඩම් ටිකක් කියන්න පුළුවන් නේද? Likewise they're differently their parts, right? අනිවාර්යයෙන්ම ionization energy bar definitely they check the electronegativities කියන එක දානවා කියලා bar, right? Definitely know whether you know the concept of mole කියන concept එක දානවා. ඊපස්සේ අපිට කියන්න පුළුවන් several things. Likewise in biology also here we can check කරලා අපිට කියනවා මෙන්න මේ ටික අහනවා කියලා. Definitely ask අහනවා. So for that we have kept 6 months. Before 6 months everything will be done. Empty bonds කියන එකක් නෙමෙයි මේවත් bonds build, right? Saturated means empty spaces කියලා නේද? Empty space, potential space. පුරව ගන්න පුළුවන් හැකියාව තියෙන places which can be filled, right? Then ළමයි these are actually organic chemistry. When you do the organic chemistry you will identify these things very well. But now you should know. Then in these unsaturated fatty acids, this is a concept in organic chemistry according to the way in the minimum now. Organic chemistry just for this right? In a double bond carbon double bond, if the same group you can make it a CH2 then hydrogen atoms, right? If the same group is in the same sides are taken like this, two sides of the double bond from the two sides of the double bond, if same molecule is present here in the same site within the right? So here you can see CH2 molecules are present in the same site. Such a molecule is called a cis. Can you cis type of structure here? Why it is called cis? What is the organic chemistry here, right? Cis, right? Cis. with the way that is mirror image organic chemistry teach what you want to know is if same type of molecules are present in the same site of a double bond that is called cis. If it is like this, same type of molecules are present at the different site, two different sites. Different sites can mean two sides from the double bond. You cannot trans cis and trans. That is how cis and trans get separated. Right? Let's read it and thereafter let's draw it. Cis unsaturated fatty acids contain double bonds with hydrogens on the same side. Hydrogens when known in NAD. Other molecules can also be there. Hydrogens on the same side of the double bond, which causes the carbon chain to bend. Trans fatty acids also contain double bonds, but the hydrogen are on opposite sides of the double bond. Other molecules can also be there, right? Other molecules can the same molecule. Since hydrogen is given, let's go with hydrogen. Right? Since this is trans, hydrogen right? With respect to other things also we can define it, but we are making hydrogen right? Cis trans when you learn about the organic chemistry, right? Now I will replace part of right? Menna hydrogen. Cis and trans. Right. Right, you can draw it. I'll explain. Right, screening right. Then in the book it has been given touch of point. I have skipped that. Right? Purposely, I have not mentioned it here because we have 2023 students are there. We very recently discussed atherosclerosis. Now, by the consumption of these fatty acids, excess amount of saturated fatty acid consumption can cause atherosclerosis. That means they can deposit blood vessels, right? We have a lesson grade 12, grade 9. Fifth lesson, functioning animal structures. We learned in that one. Blood is transported in blood vessels like this. The whole of them is known LUMEN, lumen. Sidura lumen. This is the lumen. Right? Due to the deposition of these fatty acids, unsaturated fatty acids, that lumen, size of the lumen can get reduced. Red color, I'm going to show the deposition of fats. Previously, the lumen was up to this much. The lumen, you don't need to draw this. It's a lumen. But now, due to the deposition of fats, fat deposit. Several layers of fat can deposit. Then the fat deposition looks like this. So as a result of that, now you can see actively the size of the lumen has reduced. This is called the atherosclerosis. Atherosclerosis situation, right? They can deposit in our blood circulatory system, right? So with that, we finish that part. We got to see right. That is related to the cis unsaturated fatty acids. Then we have to discuss the molecule which we know very well. Phospholipids. Second thing is phospholipids. So for lipids, the phosphate group is joined. Where do we come across this phospholipid? You discussed. Even today I mentioned plasma membrane, cell plasma membrane contains of phospholipid molecules. Phospholipid molecules, bilayer, 2 layers of phospholipid molecules. So all of us, plants, animals, everybody contains this phospholipid molecule. So it is important to know its structure, how it has become, or how it possesses a hydrophobic tail, how does it possess a hydrophilic head, likewise, right? Phospholipid molecule. Phospholipids, they are composed of 2 fatty acids and 1 phosphate group attached to a glycerol molecule. Glycerol plus 3 fatty acids. 1 fatty acid is removed and instead of the phosphate group is attached. The phosphate group gives a negative electrical charge to the phospholipid molecule. Typically, an additional polar molecule or small charged molecule is also linked to the phosphate group. Example, chlorine. Phospholipids are major components of the cell membrane. Right. Now again, I'm going to draw the structure, but this structure is strictly not essential for you. We're not going to structure. So I'm going to limit this structure just to a it's too much, unnecessary for you. So I'm just going to draw the structure. Right? The structure. Right? We structure on there. Don't need this structure. But how this is composed of, right? Glycerol මේ පැත්ත රඳින්න ඕනවා. එකවරම කරන්නේ. Do you understand this structure? මෙතන නිකන් තේරුණාද? මේ මොකද්ද? CH2OH. CHOH. CH2 මෙයාගෙන ගන්නවා. මෙතන නිකන් glycerol molecule. This is the glycerol molecule. Did you understand? තේරුණාද glycerol molecule? glycerol molecule. Right? Then here methane had R group hydrocarbon chain. So it is a chain of carbon and hydrogen. The good chain continuous chain. Chain. Single bonds CH2. And there is CH3 right? 18 was the you don't need to grow 18, right? I'm just showing you a structure. You make amino only, yeah? You don't need to draw this one. Right. So this is the fatty acid. Fatty acid joint. Fatty acids. Right. Now, previously, one glycerol molecule joined with 3 fatty acids. Then what did I tell you now? Instead of 3 fatty acids, a phosphate group is joined here. Methane joined with phosphate. But sometimes phosphate chlorine molecule could join in a pulver. Join right? This structure can change. May structure can join change with the pulver, right? Phosphate molecule is phosphate PO4 3 minus phosphate here. PO4 3 minus, right? Phosphate on a phosphate. So, right. Right. PO4 3 minus. Right? So this is the structure. Now let's compare this to the structure that we know. Compare phosphate, right? Phosphate. Phosphate. Right. On the structure, come on. All these structures, actually, all of them together are like the hydrophobic head. Hydrophobic head. And these structures are like, right? Make chains together, right? Make the make a hydrophilic head. Right. Is it clear for you? So we'll make it down. Right? Wait a minute. That's why the structure is going to be a bit different, right? May take a look at the structure, 18 chains should go down, but I think you can get the idea. These tails extend. Tails gather extended. phospholipid එක මෙන්න මෙහෙම අදින්නේ. සාමාන්‍ය ජෙට්ලයින් වල මෙන්න මෙහෙමනේ අදින්නේ. දාලා ටේකර්ස් එකක් ගන්න. The meaning. This is how it is composed. Is it clear? Here, glycerol part එකක් තියෙනවා, phosphate part is there. These are the tails. මේ tails ටික. Clear? Is it clear for you? can't understand a common තේරුම් ගන්න ලොකුට. මේ glycerol molecule කියන එක තේරුණාද? මම ඊට structure එකක් දෙනවා. I'm going to give you a structure. This is glycerol, may phosphate එක, right? And this is fatty acids. We very recently discussed. අලින් කතා කරාට කොහොමද උනේ? ඊට පස්සේ කියලා දෙන්න. Some of you understood okay. Come on, මේක අහන්නේ. ඊළඟ පොතේ දාලා is not task. But you should understand this. මේක නම් තේරෙන්න. මේක මෙතෙන්ද. තව තේරුම් නැති කට්ටිය නෝද දාන්න. glycerol molecule එක මෙහෙම ඇන්දොත් තේරෙනවද ඔයාට? Glycerol molecule. Previously, it was joined with 3 fatty acids. 1, 2, 3. මේ මොකක්ද? glycerol, fatty acids. මේ මොකක්ද? What is this? මේක දැනගන්න. Usual triglyceride. Usual triglyceride. Now, what is the difference on the key? Huh? What is the difference? Phospholipids. They are composed of 2 fatty acids and 1 phosphate group attached to 1 glycerol molecule. Reread. They are composed of 2 fatty acids and 1 phosphate group attached to 1 glycerol molecule. Instead of this fatty acid group, what has happened? The phosphate group joined. Phosphate. Now phosphate kill the PO4 minus phosphate group. Ocharaila. This is this one. Make and then mass will be found. When you do organic chemistry, you can write. But for those who can understand, come up. You understood what's going on. In the glycerol molecule, I have drawn parallel. Yeah, parallel. Is the glycerol molecule? Is the fatty acid part. Fatty acid part with the CH. You go right? When you keep on drawing it, it will be really simple for you. Right? Maybe in a tail like a fatty acid. R denote. This is the R part. R long chain. They become the tail. Right? This is the newly joined phosphate part. Phosphate. With a chlorine could pass a joint. Chlorine, no what is it? Did you understand this one? Make it. Draw this one in the image part that I have given to you. The image part. This is the structure, the way how phospholipid is composed of. Thank you. Biology. These are simple structures. This is make it. Xantica gold there the mole malakada came remove and obviously consciously is it to be structures. Maybe organic chemistry, but you will have to go across these things in chemistry. Right. Still, in the space that is given here, you should draw this one. Make with Rakha. At the first space, sorry, phospholipid molecule second space. Phospholipid. Image I'll give you that image, right? Phospholipid bilayer image. First cage is to draw paste that image. Second cage is to draw this phospholipid molecule. How is it? Did you understand? Is there anybody who didn't understand this structure? I'm talking about this one. Those who understood this one, it's good, not a difficult one. Well, then glycerol again me and they right? At least this one, did you get this one? Right? Instead of one fatty acid, the phosphate molecule join. And I got to quickly draw it. Okay, no problem with that one. Then let's talk about the functions of lipids, right? Lipids are the functions. Food reserve as energy source, right? So it also can store food as an energy source. Triglycerides such as fats and oils, right? Maintain the fluidity of plasma membrane. Actually, we have to discuss about the plasma membrane now. Fluidity means it's like this. If I take several pens like this, right? Plasma membrane actually it's not a 2D structure, it's a 3D structure, right? So in the plasma membrane, phospholipid molecules are arranged like this and they can move. See what happens here when I'm doing this. take a look at meaning of maintaining fluidity, if you take a look at these panes now, right? I'll take this side, right? Now you can see, right? When I am doing this, the positions of the panes, right? They get changed. Likewise, these phospholipid molecules can change their position. They can change their position. That is known as the fluidity of the plasma membrane. We have to talk about that later under the plasma membrane. For now, that's enough for you to remember, right? Maintain the fluidity of the plasma membrane, phospholipids and cholesterol. Let's see how that is happening and all that in the plasma membrane. Later, we have to discuss those organelles. Act as signaling molecules, example, hormones that travel through the body. Yes. Hormones, most of the hormones are lipids. So they send signals. Follicle stimulating hormone sends a signal from the pituitary to the ovules, right? Follicles, and then grow in FSH. There's a luteinizing hormone, then sends another signal. Start the bursting, burst wind, release the egg release current on the messages, right? Growth hormone sends signals throughout the body. Grow in, increase, right? Ready wind. Likewise, hormones send signal. You know what are hormones, right? You know, right? Found as components in animal cell membrane. Cholesterols, not only the phospholipid molecules, cholesterols are also found. Cholesterols are also lipids, right? So those are the characteristics of the things we have to discuss about lipids. In the next class, let's talk about the proteins. Again, proteins are not a lot of information is there, but amino acid breakup, right? Then the bond which is formed between amino acids we have discussed, right? Peptide bond, we call it as a peptide bond. Then something you didn't learn in the previous classes, structures of proteins, right? Structures of protein should be taught to you. Protein structures, right? Very important one, right? Protein, right? Amino acids. Within the next 2 weeks, we are going to finish the initial part of that one. The part, right? Any question from the things that I discussed today? Any question? Or did you understand all of them well? Disturbances, right? Disturbances are here and there. I know the condition then say take so that live. Right now I activated this systems maximum amount. Right? Any question? Then my messages are for you. Those who are in the live class, most of you may not need the recordings, but get the recordings. Again, still, if you read the Campbell biology book, it's okay. Campbell biology read it, right? But you know the importance of O-level books. O-level, please read the O-level books over and over. O-level science book. Here, be well versed of the O-level science book, right? So those are the messages I have to give you. If you don't have any question, I'm going to wind up the session. And how to identify whether hormone is protein or lipid. The simplest thing, as you know, do a protein test. Protein test. Then if it is protein, it will respond to that one. Copper sulfate and sodium hydroxide. If it is lipid, that will respond to lipid test. Sudan 3 or Sudan 4. Simplest. But there are a lot of other methods. The methods, active components. If it is composed of amino acids, protein, right? Amino acids theoretically check. A lot of other tests are there, right? A test but as you know, right? Explain about the hydrophilic and hydrophobic, right? Hydrophilic means loving water, can combine with water, likes to combine with water. Vaturatika join when a kamati. Vaturatika join when a kamati. Likes to join with water, right? Hydrophobic means doesn't like to join with water. Alright, in that tute. Phospholipids The phosphate group gives the negative electrical charge to the phospholipid molecule. Like that PO4 3 minus. That's why I have drawn it like that also. So this phosphate molecule, right? So there is a negative charge due to this one. Negative charge. That's it. Huh? Typically, an additional polar molecule or small charged molecule is also linked to the phosphate group. Example, cellulose, right? Phosphates are major components of the cell membrane, right? So here, this head, make a head including glycerol and phosphate, hydrophilic, likes to combine with water. The tail, which is composed of hydrocarbons, RCH, that is hydrophobic, doesn't like to combine with water. Waturat ekka gෑවෙන්න සම්බන්ධ වෙන්න කැමති නැහැ. Hydrophilic and hydrophobic names in name. Right? ඔය ජල වීදිකාව කියලා එකක් තියෙනවා නේද? වෙනද වෙන්න. It spreads from the bite, so at the saliva in the microorganism කෙනෙක්, right? Dog bites. මල්ලෙක් කෑම හැදෙන්නේ ජල වීදිකාව එකට කියනවා hydrophobia කියලා. Phobia, බය. Hydrophobic, වතුරට බයයි. Right. Tube now you can start writing of the tubes, right? I'll be starting the discussion as soon as the first lesson is finished. Lesson discuss so that we can recall the first step. If I discuss the tube now, I think make a carbohydrates them matakai. After 2 or 3 months, you forget these things. Then when I'm going with the tube, I tube to discuss then you can recall these things. That's why tube is taken a little bit later. It is something done purposely. Revision. It's going to be your revision. Physical classes this will continue, right? But I mean candy, right? Physical classes candy, but worry when the online classes are continuing. Then 2023 1.5 years online class continue. No any problem. No any problem. Very well. Right? Introducing new things, right? Zoom update with that one biology is later, 5th lesson right? It becomes under the online classes are continued with all the facilities. Introduce right? I should also change my clothes. But somehow, when I erase the board, when I erase the board is last right? Very easy to explain the diagrams, right? How easy diagrams keep explain make a meaning from the animal, right? Right, totally. I should wear a dark color shirt or dark color shirt. Right? Now you can very easily explain it. Very easy, right? Becomes so easier for me to explain the diagrams. Right? These sort of diagrams are not here for your lessons. Maybe one part of it. But when those diagrams appear, can very nicely, very easily explain you all the parts, right? Maybe this is that part, this that part, maybe it goes like this, goes on like this. How this process occurs like this. Your facilities are used, right? I mean, background effectively used. If I wear a dark color shirt, dark color, right? So likewise, a lot of new things are there. A lot of new things are experimented in the online platform. You can do on the physical class now. Right? Effectively, hard take explain the road, and hard take right? chambers, blood pumping. Even the video can be shared. උදාහරණ animation එකක් දාන්න පුළුවන්. දාලා ලස්සන ඔක්කොම කරන්න. වැඩ ගොඩයි. වැඩ ගොඩයි. ඉන්නකො තව ටිකක්. Right. Wait. Okay, then with that I'm going to wind up the session. Right. Thank you very much and have a good night. මම බෙල් එකක් වගේ විතරයි පේන්නේ. Right. Okay. Okay, then thank you very much and have a good night.